As you know, all living things are made up of cells. Some organisms are unicellular, or made up of a single cell. Others, like us, are made of trillions of them. Multicellular organisms are made up of somatic cells and gametes. Somatic cells are the cells that make up organs and tissues. Gametes, on the other hand, are sex cells, like sperms and eggs. Somatic cells are able to make genetically identical copies of themselves through a process known as mitosis. We'll talk about the gametes in more detail another time because they are actually formed through a different process known as meiosis. Mitosis consists of 5 parts: prophase, prometaphase, metaphase, anaphase, and telophase. First, we'll take a closer look at prophase. In early prophase, the cell starts to condense its chromosomes so that they will be easier to move. A mitotic spindle made of strong fibers known as microtubules begins to form between the centromeres as well. This will later help to pull the chromosomes to opposite ends of the cell during division so that each new cell will have the right amount of genetic information. Additionally, the nucleus starts to break down, giving the mitotic spindle access to the chromosomes. That brings us to our second step, prometaphase. Prometaphase is somewhat of a continuation of prophase. In prometaphase, the chromosomes finish condensing and the nucleus completely breaks down. Microtubules from the mitotic spindle then attach themselves to specific binding sites known as kinetochores on the centromeres of sister chromatids. But then, what do the microtubules that don't connect to chromosomes do? Though they may not have a sister chromatid to pull, these microtubules are still useful for division. They are able to use each other to stabilize the mitotic spindle. Next is metaphase. I like to think of the M in metaphase as middle, because in metaphase, all of the chromosomes line up in the middle of the cell. Now it's time for cellular tug-of-war, also known as anaphase. In anaphase, the microtubules connected to the chromosomes shorten, while those that aren't elongate. The proteins holding the 2 sister chromatids together are also weakened. This allows the mitotic spindle to pull apart the sister chromatids. Some people find it easier to remember the A in anaphase for apart or away, because the 2 sister chromatids are moving in opposite directions. Finally, the cell undergoes telophase. Telophase is almost like reverse prophase. The mitotic spindle starts to break down and 2 new nuclei begin to form. Since they are done moving, the chromosomes start to loosen up and become stringy again. Now, this almost looks like what we started with, except for one problem. The cells are still connected. To separate, they must undergo cytokinesis. Cytokinesis is not really considered a step of mitosis because mitosis deals with the division of the nucleus, while cytokinesis is the division of cytoplasm. In animal cells, actin proteins close in on the center, creating what is known as a cleavage furrow, which eventually pinches them off into 2 individual cells. Plant cells, on the other hand, cannot do the same because of their rigid cell walls. Instead, they form a cell plate down the middle and then split on either side to form individual cells. That's all for this video. I hope that you learned a lot, and if you're interested in learning more about cell division, I will be making another video soon to explain meiosis.